So the most recent research coming out of my lab was regarding what happens to hematopoietic stem cells as we age, and specifically what happens to how much they're able to produce progenitors by exiting from quiescence, becoming active, and then differentiating. And we discovered that through analysis of multimodal single-cell genomics data, we discovered a new gene regulatory network that controls that process of how stem cells exit from quiescence and start to proliferate and then go back into quiescence. And the second part is really important because that's what starts to go wrong as we age. They're no longer able to slow themselves down, and that leads to aging phenotypes. So I run a theoretical lab and I collaborate with researchers here at FLI, and so we do the data analysis using both methods that exist, and also we generate new computational and mathematical tools. So in this case, we really wanted to understand how genes interact and study gene regulatory networks, and we did that by developing a new method for gene regulatory network inference. So which are the most important genes that control the transcription of other genes that are regulating stem cell proliferation. In this project, in addition to the main tools that we use for gene regulatory network inference, which we developed, we're using data generated by the Rudolph lab, which is single-cell multi-omics. So they're sequencing both gene expression and chromatin accessibility in the same cells. And on my perspective, some of the great excitement I have in working with these data is I come from a background of mathematical modeling. So we're using the methods of fitting these kind of genomics quantities into dynamic models that we can then simulate and look at how stem cell differentiation changes over time or between different lineages of the blood system as we age. And that's the kind of the strength of mathematical modeling in that area. Well, I think right now we're looking at quite fundamental properties of the hematopoietic stem cells, like how well they're able to proliferate or stay in a quiescent state. But of course, to be able to manipulate that would be powerful because that would let us have more control over the health of our stem cells as we age. Because one of the great challenges is that the output of these stem cells, their functional quality starts to decline as we age, and we think a part of that is in the case of the hematopoietic stem cell system, an inability of the cells to go back into quiescence. So ultimately the added value would be an ability to kind of slow down the functional aging of the hematopoietic stem cells and be able to maintain a healthy blood and immune system for many more years. My connection is, so this project that I described is a collaboration between my lab at USC in Los Angeles, and we do all of the computational modeling, and the lab of Professor Leonard Rudolph here at the FLI, and his lab has conducted all of the experimental work. So that's my connection, that's why I'm here, and it's been a really great collaboration. Being a theoretical biologist, I always I don't generate data myself, but I'm passionate about kind of being close to the data for both myself and my students. So I think the projects that are really successful as collaborations are projects in which theoretical and experimental biologists are working together throughout, not just at the end, like after the data is generated. So I think that's really been a strength of our project, is that we've worked together as labs for a few years building up to this in all the phases, from the experimental design to the generation of data, and then to the analysis and the computational modeling and the insight that we gain.